In our next screencast on Chapter 9, we're going to go into Phase 2 of cellular respiration, which is the citric acid cycle, aka the Krebs cycle. All right, so we've just come through glycolysis, okay? Glycolysis, again, if you remember, occurs in the cytosol and is anaerobic, and takes one molecule of glucose and breaks it apart and yields two molecules of pyruvate. Okay, so after glycolysis is finished, if oxygen is present, okay, then pyruvate will enter the mitochondria and the oxidation of glucose is completed there in a series of two more major steps. Okay, so after we oxidize pyruvate, citric acid cycle completes the energy yielding oxidation of organic molecules. So after the citric acid cycle, all of glucose is broken down and all the energy is released. So let's go through that. So once we have pyruvate in the cytosol, and we have to get pyruvate into the mitochondria. And if you recall, mitochondria have two membranes. And if you recall everything from what we know about getting things in and out of membranes and across membranes, it is no easy trick. So we have to get pyruvate into the mitochondria. Part of that process continues to metabolize pyruvate and converts it into something we call acetyl coenzyme A. Okay, and the conversion of pyruvate to acetyl CoA links glycolysis to the citric acid cycle. Okay, this step is carried out by a multi-enzyme complex called the pyruvate dehydrogenase complex. Okay, and again, because it's dehydrogenase, we know that it's going to reduce some NAD+. Okay, so let's watch that. So here we have our pyruvate in the cytosol. Okay, and you'll notice at this point, pyruvate is still a 1, 2, 3 carbon molecule. It's still half of glucose. And again, also remember that everything from this, because I have two molecules of pyruvate from the end of glycolysis, everything from this point on is going to be doubled, right? So as pyruvate is transported into the cytosol, the first part of the pyruvate dehydrogenase complex removes a molecule of carbon dioxide, and that comes from this right here, okay? In doing so, it releases some high energy electrons that are used to reduce NAD+ into NADH plus H plus. Okay, so, and as you remember, these NADHs are going to go to the electron transport chain and carry those high energy electrons there so that they can be used to synthesize ATP. Okay, the third step is the addition of coenzyme A where that carbon dioxide molecule was. And if you look, this part of the molecule is the same, but that carbon dioxide is replaced with coenzyme A. And so at the end of this process, we have a molecule called acetyl CoA, which is this acetyl group right here, and a molecule of CoA. And now acetyl CoA is the substrate for the citric acid cycle. So now let's have a look at the citric acid cycle. It's also called the Krebs cycle, and it completes the breakdown of pyruvate into carbon dioxide. Now, conversion to acetyl CoA has already released one molecule of carbon dioxide, and so there's two carbons left on each pyruvate. The citric acid cycle uses organic fuel derived from pyruvate. So the organic fuel derived from pyruvate, as you remember, is acetyl-CoA. Okay. And as it goes through the cycle, it generates 1 ATP, 3 NADH, and 1 FADH2, which is another electron, an alternate electron carrier. It's alternate to NAD+. Okay. Per turn. So it generates all those per turn. So remember, we have 2 molecules of pyruvate, so all that will be multiplied by 2. This is a quick summary of what happens. So here is the conversion of pyruvate to acetyl-CoA. Okay, and again, we're going to multiply everything in there times 2. And then the citric acid cycle, as we go around, everything in here is also going to be multiplied by 2 for each molecule of pyruvate, 2 pyruvates. And then this just shows kind of the highlights of the citric acid cycle. Okay, over the course of the citric acid cycle, 2 molecules of carbon dioxide are produced, 3 molecules of NADH are produced when we reduce NAD+ with high energy electrons. We make 2 molecules of ATP, and again, all this is times 2, times 2. And then some FADH2, which again is an alternate electron carrier. So I'm just going to go into next a little bit more detail on the citric acid cycle. So here's a little bit more detail on the citric acid cycle. The citric acid cycle has a total of 8 steps, and each one is catalyzed by its own specific enzyme. So this pathway is 8 steps, and each one has a different enzyme. So the acetyl group of acetyl-CoA joins, which remember, the acetyl group is what's left of pyruvate. So that's where all the energy that remains from glucose is still stored. So that acetyl group joins the cycle by combining with something called oxaloacetate. And then that forms citrate, and citrate is the first product of the citric acid cycle. Okay. The next 7 steps decompose citrate back into oxaloacetate. So that's what makes this process a cycle. And you'll see this in a minute as we look at it in more detail. Okay. And then when we reduce our electron carriers, we generate NADH and FADH2. They carry our high energy electrons to the electron transport chain, which is where the bulk of our ATP will be made. All right, so let's walk ourselves through this. So now we have acetyl-CoA here, and remember, this acetyl group is what remains of pyruvate. So that's where the remaining energy of glucose is stored. That acetyl group combines with oxaloacetate, okay, and then forms. So you see, if you look at the color coding, so we've got 2 red carbons here and 3 the blue carbons here. And there's that acetyl group right onto oxaloacetate to make citrate. Okay. Then in the next step, okay, you have a little bit of exchange of some water in, some water out. There's a little bit of a rearrangement, and you get isocitrate, which is an isomer of citrate. The next step is catalyzed by an enzyme called isocitrate dehydrogenase. And because it's a dehydrogenase, okay, it reduces a molecule of NAD to NADH. So there's the first of our NADH made. And we're left with alpha-ketoglutarate. And so, and it also releases a molecule of carbon dioxide. Okay. The next step, we take alpha-ketoglutarate. Okay. again, another dehydrogenase enzyme here. We liberate another molecule of carbon dioxide, okay, and reduce another NAD into NADH. So there's our second NADH. And the product of this step is called succinyl-CoA. In the next step, succinyl-CoA, let me move this up just a scotch so you can see it there. Succinyl-CoA is converted into succinate, and as it does that, it takes a molecule. It provides enough energy to phosphorylate GDP into GTP, but that GTP is not our favorite energy source. So it uses its energy to convert a molecule of ADP to ATP. And again, this is all substrate level phosphorylation. Okay, so now I've got 2 molecules of NADH, 2 molecules of carbon dioxide, and 1 ATP. However, we're not done wringing the energy. Think about, you got to wring a towel, wringing the energy out of our organic molecules. Okay. Then we take succinate and convert it to fumarate. In the process, we release a little bit more energy in the form of high energy electrons, and it reduces FAD, which is our alternate electron carrier, into FADH2. Okay. And then we have fumarate. It is converted to malate, okay, in the next step. And then the final step, which you'll see, is basically malate is converted into oxaloacetate. And the last little bit of energy is squeezed out, reducing NAD plus into NADH. Okay, so now there's the entire cycle down there in front of you, and I'm going to go around and count up. And again, you don't need to memorize all of these intermediates and all of the phone and all of the, you guys didn't need to see that. You don't need to memorize the intermediates or the enzymes. So, but you do need to know, you need to be able to kind of basically follow the flow around the cycle. You need to know that it is a cycle. So the first thing we use is the last thing that's generated. And you need to know kind of what steps involve our energy. So up a little bit, there, and there, and there. Okay. So again, over the course of the cycle, remember this goes around twice because there are 2 molecules of acetyl-CoA that get fed in. So we produce 2 NADH in this step and 2 molecules of carbon dioxide, 2 molecules of CO2 there, and 2 NADH, 2 total ATP, 2 FADH2, and 2 NADH there. So for our grand total from the citric acid cycle, we've got, not 2, 4 molecules of carbon dioxide, okay, 6 NADH, and 2 FADH2, and 2 ATP, okay. The carbon dioxide are put back in your bloodstream and you breathe it out. It makes its way to your lungs and you breathe it out. The NADH and the FADH2 will be taken to the electron transport chain in the next step of oxidative phosphorylation, and they will give their high energy electrons to the electron transport chain so that it can fuel the synthesis of ATP. And we have 2 ATP, and those 2 ATP just hang around and wait to be used for cellular work. Okay, they are ready to go. All right, I know that's a lot. Go through it a couple of times. We can talk about it in class, but that is pretty much it on the citric acid cycle. The end.